Welcome, everyone, and thank you for joining us for today's webinar, Synthetic Antibodies, the Emerging Field of Aptamers in Diagnostic and Drug Discovery, presented by Dr. Thomas Caltagirone, President and CEO of Aptagen. My name is Dr. Susie Valdez, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I want to remind everyone that today's event is interactive. We encourage you to participate by submitting as many questions as you want during your presentation. Just simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have any trouble seeing or hearing this presentation, just click on that Ask a Question box and let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credit tab located at the top right corner of your presentation window and follow the process for obtaining those credits. Without further ado, please join me in welcoming our presenter today, Dr. Thomas Caltagirone. Welcome, sir.
Thank you, and thank you for that introduction, and thank all of you for attending the talk today. As mentioned, my name is Tom Caltagirone. I'm President and CEO of a biotech company called Aptagen. For this talk, I hope I'm able to not only educate, but inspire some of you to consider the technological possibilities of using aptamers in a wide variety of fields and downstream applications. Our bold and in-your-face mantra is simply forget antibodies, use aptamers. So I started the company back 15 years ago. In 2004, we are a service provider. We're located in South Central Pennsylvania, and we have clients ranging from startup biotech companies to research academics to big pharma like Pfizer and Merck. They approach us to develop aptamers for one reason, and that is either their antibodies failed to specifically target their antigen, or the antibody doesn't exist whatsoever. So against difficult targets or antigens. So they're considering alternative platforms like aptamers in their downstream applications. So I'd like to segment this talk into 4 sections. First, I'll introduce aptamers, background of aptamers, diagnostic applications, cell and tissue targeting and delivery, and also drug discovery applications. So what are aptamers? Here's a short review paper that we published. Aptamers are sometimes referred to as synthetic antibodies, but they're not protein-based macromolecules. Actually, I brought along this prop to illustrate the point. If this represents a single-stranded DNA or RNA molecule, depending on the sequence, it will fold into some sort of 3-dimensional structure. It's still nucleic acid. But once it binds to a target, we call that an aptamer. So they were invented back in the early 90s. And targets can range from small molecule targets to protein biomarkers, cells, tissues, viruses, you name it. And they operate in a wide variety of matrices, including biological fluids. So here are some examples of aptamer structures, or back in the early 90s, of success stories of aptamers against a wide variety of targets, X, Y, Z, whatever that may be. And here are some examples. For example, there's a pseudonaut configuration against HIV reverse transcriptase. There's an aptamer that takes the form of a G quadruplex that targets thrombin. There's a simple hairpin aptamer against T4 polymerase, and a stem loop bulge against ATP, a small molecule ATP. So these are just some simple structures of early examples of aptamers that have been developed over the years. The literature has exploded in terms of success stories in the last 6 years of aptamers targeting a whole host of targets and antigens. The size of aptamers are approximately 1/5 the size of a typical IgG antibody, about 30 kilodaltons. And because of its smaller size as a macromolecule, it's much more advantageous to have a smaller size versus a bulky antibody in terms of penetrating tissues for targeted therapy. Also, for biosensing applications, if we tether or couple the aptamer to a biosensor surface, because it's smaller macromolecule instead of a large bulky antibody, your signal response on your biosensor is much more improved. So the biosensor community typically love their aptamers over their bulky antibodies on their biosensors. Okay, in terms of their biophysical characteristics, affinity. Aptamers have comparable affinities to that of antibodies, but where aptamers outshine antibodies is in specificity. So affinities can be in the nanomolar range, low nanomolar range, depending on whether it's a large target like a protein, and in some cases a picomolar. But in terms of specificity, they are much more specific than antibodies. You can minimize or eliminate cross-reactivity or false positive results of using aptamers versus antibodies, simply because of way of how they're developed, and I'll get into that a little bit later in the slide deck. But here's an example of an aptamer that exists against theophylline versus caffeine. Two small molecules. Typically, small molecules are difficult targets for antibodies, but there's an aptamer that binds to theophylline versus caffeine with 10,000-fold greater specificity, and they only differ by a single methyl group. So just a few atoms, and yet this aptamer binds to theophylline versus caffeine. I don't think you can do that with an antibody in most cases. Also, another advantage of aptamers over antibodies is that you can discover unknown or unique biomarkers in biological samples. For example, you can, if you have a patient sample or samples that are considered positive for some pathology or some disease by some clinical means, and you have another set of patient samples of a biological fluid that is considered negative, we can develop aptamers that respond to the positive and not to the negative. And so once you have that, you can easily identify what the biomarker is by performing a pull-down assay and mass spec analysis to identify what the biomarkers are. So aptamers have that advantage to discover unknown biomarkers that exist in biological samples and patient samples. In terms of manufacturing cost or in production, they're a fraction of that compared to antibodies. So I typically say pennies on the dollar in terms of producing or manufacturing aptamers versus antibodies. Also, aptamers are much more stable than antibodies. They have a longer shelf life. So because they're nucleic acids, DNA or RNA, you can, back to the model here, you can heat denature them, cool them down, and they'll snap back into shape. You can't do that with a protein-based antibody. turns into scrambled eggs after you heat it. So aptamers have that benefit as well in terms of the longer shelf life. So also have listed here on this slide is what we have displayed on our website called the Apta Index. It's a searchable database that we provide to the scientific community to see if an aptamer exists. You simply type in your target or antigen of interest, and we can provide the sequence of that aptamer, the DNA sequence or the RNA sequence of that aptamer if it exists. Now, it's not an exhaustive list. This is information that we pulled from the literature, and again, this is a resource to the scientific community that we provide on our website. We're in the business of developing custom aptamers, but this is a resource to see if an aptamer does exist against your particular target of interest. So, diagnostic applications, switching gears here in terms of diagnostic applications. So we're all familiar with the typical sandwich ELISA method using antibodies. It's the gold standard. It's a time-consuming, multi-step approach using primary and secondary antibodies. There are incubations, washes, capture steps. It takes hours to complete. We can develop aptamers, sandwich-based aptamers, for this type of sandwich-based assay. However, we have developed something that's superior to that of ELISAs or sandwich-based assays called aptabeacons. And these are aptamers that, upon binding to a target, reports an output, either a fluorescent output or colorimetric output. So in this cartoon diagram on the left panel, you see a generic aptamer structure, a simple hairpin, let's say. On one end of the aptamer, there's a fluorescent tag or fluorescent probe in close proximity to a quencher. So in the presence of the negative sample, or the sample that doesn't contain your antigen of interest, no reaction occurs. However, on the right-hand panel, in the presence of your positive sample, the quencher is released and you exhibit a fluorescent response. And this is a one-step, one-pot reaction, and free in solution. Unlike ELISAs or sandwich-based assays, there's no capturing, there's no washing, you just simply read. And we call these aptabeacons. On this slide, and there's aptabeacons is an overreaching concept. And there are different versions of aptabeacons. In this particular version, it uses an allosteric ribozyme switch. So in the top left diagram here, it's an RNA molecule. There is a hammerhead ribozyme motif. The arrow, that little arrow that you see on near the quencher strand, is the cleavage site. So this is a naturally occurring self-cleaving ribozyme that exists in nature. Juxtaposed to that is the aptamer domain, where the effector binding pocket occurs. So in other words, if you have a particular target or antigen that the aptamer recognizes, it binds to that pocket, and upon binding to that domain, it causes an allosteric conformational change that activates the self-cleavage event, releasing the quencher strand and thereby exhibiting fluorescence. So this is an allosteric riboswitch with fluorescent quencher tags, and this is one version of the mechanism or the design of an aptabeacon. There are other versions. In fact, we've exhibited this or showcased this in a demo kit where it's actually a colorimetric reaction. There is a fluorescent version of this demo kit as well, And this targets, as a demo, this targets theophylline, the example I gave earlier, where theophylline, the theophylline sample turns blue, unlike the caffeine sample that remains negative and clear. But this platform, this diagnostic platform, can be applied to virtually any target, small molecules like theophylline and caffeine, or protein biomarkers, cells, tissues, viruses, you name it. In that kit, this is actually, it uses that allosteric ribozyme mechanism that I mentioned earlier, where on the left-hand panel there's the off state. It's a shape-shifting switch, if you want to call it that way, or actually an apta-switch. When it's labeled, we call it apta beacons, but in its naked version without the fluorescence, we call that apta-switches. So in the presence of the antigen or analyte, the apta-switch turns on, releases an initiator or trigger strand that initiates a cascade of events, amplification events, to enhance the signal. So this kit entails a signal enhancement system to boost the signal for sensitivity. So there's 2 levels of amplification before final color development. We recently published this in ACS Biochem, and this is the actual nuts and bolts of that one-pot reaction, or this 3-stage signal enhancement system. On the left-hand side is the allosteric ribo switch in the presence of target. Again, we've studied, we've used as an example, as a generic example, theophylline versus caffeine. The trigger that's released after the reaction takes place initiates an XPAR reaction. This is an isothermal amplification reaction, similar to PCR, but isothermal conditions. So it amplifies that signal through this isothermal amplification reaction, generating multiple or many copies of a DNA zyme. This is a DNA aptazyme, DNA zyme. There are many different terms you can use, but this particular DNA zyme has peroxidase-like activity. So like similar to HRP, horseradish peroxidase. So in the presence of TMB on the right, on the 3rd stage of this signal enhancement system, the common substrate for a peroxidase type enzyme, it converts the reduced state of TMB to an oxidized state, and this turns into a blue color. So it's a colorimetric version. So this is a one-pot reaction. Again, it's one version of our apta beacon platform, where there's a signal enhancement system, free in solution, one-pot reaction. We can also develop these apta beacons, or convert them into, we can reconfigure them into different platforms in terms of lateral flow for point-of-care use and point-of-care diagnostics. So lateral flow test strips, like the dipstick assays in pregnancy tests, we can use our aptamers in that configuration, or apta-switches, to test for analytes. So that's another platform to replace antibodies, because typically antibodies are used in those platforms. And there are groups, research groups, that are developing smartphone readers for these, whether you're using a lateral flow that has a fluorescent output or a colorimetric detection system. So a smartphone reader that can read a fluorescent apta beacon, for example, we can employ that, or even a colorimetric reader for yes/no answers, or even semi-quantitative readouts. This slide here is a table of different targets and aptamers that have been developed against those different targets and antigens. Most of our customers and clients, they're hyper-focused on affinities. Can we develop a tight binding aptamer? And yes, we can. But affinity is not necessarily a prediction or predictive measure of success for your downstream application. That's one measurement that we take into consideration as we quantify our aptamers. You can have a high affinity aptamer or any molecule ligand for that reason, but it's not specific. So it's not selective. So the other thing that should be of concern is how specific is it to minimize a cross-reactive and giving out false positive results. And also the limit of detection. The platform that you're using, what kind of limit of detection or sensitivity are you achieving? You could have a high affinity aptamer and yet fails your downstream application. For example, these aptamers are polyanionic biopolymers. They're very sticky. And if you have a positively charged target, it will bind to any positively charged target with high affinity. So the trick is to develop a ligand, an aptamer ligand, that is not influenced by those electrostatic interactions and is specific to the identity of target and not its charge. So depending on the matrix and the platform detection, those are the factors that we consider in terms of the design and execution of the development of our aptamers. Another example, a theophylline. It has a mediocre affinity. It has micromolar affinity. It's not nanomolar. It's micromolar affinity. But yet, I mentioned earlier, it has 10,000-fold greater specificity towards theophylline versus caffeine. And you can see here on this table, for example, the one target Plasmodium lactase dehydrogenase. It's a protein target. An aptamer has been developed against that target with 0.35 micromolar affinity. So a high nanomolar affinity. But it has 0.5 femtomolar sensitivity. So depending on the platform that you're using in terms of detection, if it's a validated platform, the rule of thumb that I usually give is that whatever the affinity, the sensitivity or the low limit of detection is at least one or two orders of magnitude in terms of sensitivity. So affinity is one factor that we how tight does the ligand or aptamer bind to the target, but you also have to consider your specificity to minimize cross-reactive false positive results, and also your platform in terms of detection, in terms of low limits of detection. So those are the other parameters that we want to take into account as we design the development of aptamers. Okay, so switching gears a bit, now I'll delve into cells and tissue targeting and delivery applications. But before I get into that, I need to take a step back and introduce a concept called directed molecular evolution. It's basically using principles of Darwinian evolution, but at a molecular scale. So let's say in this cartoon representation of the process, we have a population of molecules or a library of molecules of different shapes, here represented by triangles, squares, and circles. And immobilized on a solid support, like let's say a column, we have our target or antigen of interest. And so the idea is that we want to interrogate this library, find out, fish for those for that needle in that haystack that meets a certain fitness test. In this case, binding to the target, like in a lock and key type fashion. So if there are shapes in there that binds to the target in a lock and key fashion, those are our binders, our winners that we want to propagate for another generation. So we collect those binders, and granted, the first random library, there's going to be one or more, a very few percentage of molecules that meet that criteria or that fitness test. And so the vast majority of the library are failure shapes in this example, and so they will be discarded. But the rare molecules that do exist that bind to the target are collected and propagated. And we can introduce some sloppiness to the process, introduce some mutation, slight mutation, to explore different contours of shape to enhance its binding ability towards the target of interest. So after multiple generations of this process of screening, partitioning binders from the non-binders and amplification, we enrich this library from a random state to a more homogeneous state, a polyclonal state, if you will, that has a response to the target. So this is the basic concept of directed molecular evolution. Again, we're using principles of Darwinian evolution, but on a molecular scale, and we can apply this concept to oligonucleotides, single-stranded DNA or RNA. So these molecules are synthesized on an oligosynthesizer, a DNA machine, an automated DNA machine, to generate a library of 10 to the 14th, or 10 trillion different randomized molecules. So 10 trillion different randomized sequences. Each one will fold into a unique shape. And so we're exploring that library, interrogating that library against the target of interest. In this case, here's a generic target that I'd shown this example, this schematic flow chart here. The molecules that don't form the correct three-dimensional shape that binds to the target are discarded. The ones that do bind are collected and amplified by PCR. So polymerase chain reaction to amplify those binders. And if it's an RNA library that we're exploring or interrogating, then we're using RT-PCR, reverse transcriptase or reverse transcription and PCR. So after multiple generations of this process of enriching the library, we can clone and sequence and test individual candidates in terms of affinity and specificity. Now, to eliminate cross-reactivity to the components in your matrix, whether it's a blood sample, saliva sample, urine sample, or even counter targets that are similar to your target biomarker, but are considered counter targets, we would introduce a step here where we would expose the library first to that matrix or to that environment or to any counter targets, and anything that binds to that, any of the components or the milieu of components in that sample will be discarded. What's left over is then exposed to your target. So in that fashion, we ensure that we're enriching for molecules that are specific to your target. So there's a positive selection step, and there's a negative or counter selection step involved as well. That's not shown here on this diagram. So with that simple concept, on paper it's simple, but believe me, I've been in the field for the last 25 years. There are tricks to the trade that we employ to ensure success. We have a pretty high success rate in terms of a wide variety of targets, but we have experience in targeting small molecules, proteins, tissues, peptides, cells, you name it. And so then the next few slides, I want to show you some examples, or a potpourri of examples of projects that we've worked on that some information is redacted, but some of this data is also internal results that we've generated in our lab that's outside of our non-disclosure agreements with our clients and customers. But here's one project in particular. It's against targeting a prostate-specific membrane antigen for prostate cancer. So we know that we use a circular DNA library, and the reason we circularize the library is that we want to avoid any degradation from nucleases that may be present in this in vivo environment. And so most of the nucleases that exist in vivo are exonucleases. And so in order to protect the ends of the DNA from exonucleases, we simply just coupled it together or tied it together into a circular format. So we developed designed this library to be circular in nature, and we exposed this library to the parental cell line for our negative selection step. And any molecules that bound to the negative cell line were discarded. What was left over that did not bind was exposed to our transfected cell line that expresses this biomarker on the cell surface. And whatever bound to that is collected and amplified. And we regenerated the circularized library. So after multiple generations of negative selection, followed by positive selection, we enriched the library to respond to the positive cell line, or the transfected cell line expressing the biomarker. And in terms of analyzing the characteristics of this enriched library, we used flow cytometry. So this is, so we labeled the library with a fluorescent tag, and we went through 19 generations of enrichment. So these are the flow cytometry results against the negative cell lines, or the parental cell lines, the PC3 cells. And you can see G0 versus G19. G0 indicates the initial random pull, and G19 is the enriched library after 19 generations through that process. And there's no real shift or significant shift to the right of this diagram, flow cytometry tracing. However, when we use that same comparison on the positive cell line that express the PCMA or the PSMA biomarker, you can see there is a shift of the enriched library, the G19 library, compared to the randomized control. So we were confident that we have something here in terms of a response specifically to the transfected cell line. So that was one project against a cell surface marker. Here is another project. And we, by the way, we did clone and sequence the previous project, and obviously I won't be able to show you that data. But here's another project against an intracellular target, where we selected an aptamer that bound to an intracellular receptor, and we've quantified or monitored the enrichment of this library using real-time PCR and quantitative PCR. And here are shown the real-time PCR tracings and the melt curve analysis of those different samples. In short, what we found is that the enriched library had 100-fold more preference towards the intracellular target versus the control cells that we've tested. And we, again, further, we cloned and sequenced that library to further analyze those monoclonal aptamer candidates. But here I have a slide indicating we also looked at a labeled, the enriched labeled library under fluorescent microscopy conditions. On the left-hand panel, you see the phase contrast image of these cells versus the fluorescence in the same area. Now these are against the negative cells, or the negative cell line in this particular project, and so we don't expect to see obviously fluorescence here. So this is our negative control. However, in the positive cell line, we do see fluorescence targeting these cells that expresses this intracellular receptor. So these are just some examples of cell-based selection projects that we've entertained over the years. And now let me dive into drug discovery applications of aptamers. So as we all know, from laboratory bench to pharmacy shelves, it takes approximately 10 to 15 years to bring a drug to market and costs over $2 billion. It's a long and expensive process fraught with a low success rate, because there's only one in 1,000, every 1,000 candidates that are passed testing in animals, actually one makes it into human clinical trials. So a very low success rate, even after all those years and all that capital investment into the process. And this has been the paradigm of the drug discovery development ever since the pharmaceutical industry has been in existence for the last 100, 150 years. So, and the reason for this, why it's long and time-consuming and expensive process, is because you have to have some sort of knowledge of the pathology that you're trying to treat, and develop validated in vitro or test tube assays that you can test your drug candidates. And any positive hits you get from there, then you test in animal models, and finally into human clinical trials. So now the molecules that are tested are either derived from nature. Typically the small molecule, some small organic small molecules are either derived from nature or chemically synthesized at the laboratory bench. And again, these are tested in these in vitro test tube assays. Any positive hits you get from there, you test in human animal models that mimic the human form of the condition before clinical trials. Now, more times than not, you get negative results, and so you have to go back and from in vitro test to in vivo studies. You have to tinker with the molecule. It's a trial and error approach, back and forth, time and money. So it's a very expensive, long and time-consuming process of to develop candidates or drug candidates to market. However, what if we were to eliminate the in vitro assays and test tube assays and do drug discovery directly in an animal model? If you're able to do this, then you're one step closer to human clinical trials, thereby saving significant time and money in the process of developing drug candidates. And so what if we were able to take a library, a molecular library? So I showed examples of in vitro and cell-based assays, but let's say we go to a whole animal model that mimics the human form of the disease. Let's say it's a tumor, a cancerous tumor, and we administer this library to the animal, either a tail vein injection or some other route of administration. And in this cartoon example, we collect the area of the tissue of interest. There's a cross-section of tissue here, and there's an area within that cross-section of tissue where your pathological marker exists, your biomarker exists, that black region there. That's your cancerous region. If we take a biopsy or extract that abnormal tissue away from your normal healthy tissue and extract any ligands or molecules that are found at that site, and we can amplify those molecules. And the molecules actually that in that library that you administered, the molecules that cross-react with normal healthy tissue will be left behind. So the normal healthy tissue will act as a sink to minimize cross-reactivity or side effects, if you will, because we're using, again, the principles of Darwinian evolution, but in this case, in a whole animal model, we're taking the binders that specifically target the pathological area and leaving behind those molecules that cross-react with normal healthy tissue. So those molecules are allowed to be propagated and amplified, and then we re-inject into another animal that's mimicking the same form of the disease, and we apply this process over and over again to enrich the molecular library from a random state to a more homogeneous polyclonal state that preferentially targets that diseased area. So in drug discovery, as all of you know, delivery is always an issue. So selection in whole animal models solves the delivery issue to minimize off-target effects. So you can use your molecules that target that specific tissue, these aptamers, and use it as a vehicle to carry a toxin, right? If you have a toxin on one end, this is a vehicle that carries your cargo to that diseased area. And the case I used was cancer. So a toxin to destroy that cancer, or even a radioisotope, or even possibly a drug, a small molecule drug that failed clinical trials for off-target effects. You can possibly rescue that drug in terms of minimizing those off-target effects by using a molecular bullet, if you will, to carry that cargo to your site of interest. So you can use these as carriers to specifically target your diseased area or your pathology. Now, in terms of using the same process to develop pharmaceutical compounds or drugs in general, you need chemical diversity. Okay, chemical diversity will allow you to enrich for molecules in that library that may have drug-like effects. So not only for delivery, but also as drugs themselves. So if that's the case, in the process of whole animal selection, if this is the cross-section of tissue that I showed earlier, if there's sufficient chemical diversity, you can potentially minimize the presence of that biomarker over multiple generations. And so you can end up with an effective treatment, or dare I say a cure. So as long as there's sufficient molecular diversity, right, it's possible to use this molecular library to effectively minimize that pathological biomarker. Now, in order for this to work, there needs to be several key requirements for the selection process. First, the molecules need to be self-replicating. Aptamers are self-replicating. There needs to be an animal model that exists that mimics the human form of the disease, whether it's cancer, some sort of tumor, xenograft model, Alzheimer's disease, Parkinson's, or whatever. But these animal models must exhibit a characteristic phenotype for visualization in order to take a biopsy or to, or laser capture microscopy to cut out that diseased area away from normal healthy tissue. So in essence, it's possible to target diseases of bacterial or viral in origin, or could we possibly target other tissues or organs, the brain, possibly map the brain at specific area for glioblastoma, possibly, as long as it can cross the blood-brain barrier. So you could possibly influence behavior, enhance cognitive abilities. So the possibilities are endless in terms of using this approach of molecular Darwinian evolution, but in a whole animal, whether they're aptamers or other types of ligands, but they must meet these criteria that's listed on this slide here. So towards that aim or that ultimate goal, we have undertaken a simple experiment where we generated a library of aptamers or randomized 2' fluoro RNA, single-stranded 2' fluoro RNA library. The reason it's 2' fluoro, as we all know, RNA is very susceptible to degradation. So there is an analog of RNA in the 2' position. Instead of a hydroxyl group, there's a fluorine atom. And this 2' fluoro RNA chemistry allows this RNA to be very stable, rock solid, even more stable than DNA. In fact, it has a half-life in serum of 86 hours. So we've taken a BALP-C mouse, tail vein injected a BALP-C mouse with this library. And our negative sham control was a saline injection of PBS into a mouse that did not contain the library. We post-IV, after a certain amount of time, we sacked the mouse, collected various organs and tissues, harvested those organs and tissues, purified any RNA molecules that may be found in each tissue, and performed RT-PCR. And in this, again, this flow chart of the experimental design, we hope to see a band on a gel with the mouse that has been injected versus no band on the sham control. So this is our experimental setup to see if these molecules survive in vivo, and if we can amplify RNA species that reside in those various organs and tissues. So just a very simple question to see if we can perform those techniques. And these are the actual results from that experiment. We tested a panel of tissues, and this electrophoresis and gel run on the positive lane is our positive control. Our negative lane is the BALP-C mouse that was only injected with a saline PBS. And we can see across the panel of tissues, we did receive, just a positive signal, a yes signal, yes/no type. This is a yes/no type analysis that, across all the panel of tissues that we've tested, we did see a signal, which is encouraging, because, not only do these molecules survive in a complex biological, organism, but we're able to amplify any molecules that reside in those tissues. So then the next question is, okay, well, can we amplify molecules and enrich for molecules that specifically target one tissue versus another tissue? And so we chose lung, and below on the right-hand side, the fourth quadrant on the left, on the right-hand side of this slide, we quantitated the enrichment of these aptamers using real-time PCR, quantitative PCR. The y-axis indicates the enrichment ratio, and this is simply the amount of RNA we were able to quantify using quantitative PCR in that particular tissue relative to the total bolus injection of the library itself, okay? And the x-axis indicates the rounds of selection, or the rounds that it went through this process of administration, waiting a certain period of time, collection of tissue, and amplifying those molecules and re-injecting into another mouse. So after, you can see in rounds, and by the way, the blue bar indicates the signal from lung. The other colored bars are the signal that from the other tissues, the panel tissues that we've tested. So you can see at generation 5, 6, the signal from lung has increased from the early generations. And by the time generation 7, 8, and 9, it started to plateau. So at this point, we've determined that we've enriched the library from a random state to a more homogeneous polyclonal state that has a general response targeting lung versus the other tissues that we tested in our panel. And the signal to noise here is approximately 10 to 1. So the library at that stage in generation 9 had a 10-fold more preference towards lung versus the other tissues. So this was interesting, and so we decided to stop the experiment there and take a peek at the sequence, at the polyclonal sequence. And so we've sequenced that enriched pool, and this was back in the day when we were doing just Sanger sequencing, but now we have next-gen sequencing capabilities, so we can look at hundreds of thousands, if not millions, of sequences. But here, when we use conventional sequencing techniques, we found that the sequences were grouped into 3 different families. Down at the bottom are the consensus sequence of Group A, Group B, and Group C. And right above that are the M-fold secondary structures of these families. So that's the predictive secondary structure of each of these consensus sequences. And above that is the enrichment ratio comparing the library, the enriched G9 library, versus individual clones that we've sequenced that match the consensus sequence. And immediately you can see that the clone G9C4 is the most interesting clone to further analyze. That had the highest signal towards lung versus the other tissues. So we decided to label that particular clone, and fluorescently label, I should say, and analyzed tissue sections using in situ techniques under fluorescence microscopy. And we found that that particular aptamer clone did not bind to any of the panel of tissues. We could not see anything under the scope except for lung and liver, which is surprising. Well, actually not surprising. So lung lit up, and we did see some background fluorescence in liver as well, but we had to crank up this exposure in order to show the image on this slide here. So actually the lung is twice as bright as what you see with the liver here, because we had to crank it up twice as much in terms of the exposure on the camera. But why are we seeing residual fluorescence in liver? And there could be one of 2 explanations. The first is that there might be a common epitope where this aptamer binds to that exists both in the lung and the liver. But the most likely explanation is that what we're seeing is byproducts or degradation products. Everything passes through the kidneys and liver, and so this is the clearance of the fluorescent fragments of these aptamers through the liver. So that's the most likely explanation. Okay. So switching gears a bit, I'd like to introduce the concept of next-gen aptamers, or aptabodies, as we coin the term. These are not antibodies, and it's not an aptamer, it's an aptabody. So you can think of it as a hybrid between the two. So we know, I've talked about the, aptamers. They're single-stranded DNA or RNA. It's a naked oligonucleotide, not much chemistry besides the 4 bases: adenine, thymine, guanine, and cytosine. And we can use the aptamers to specifically target for diagnostics or as a drug discovery tool, and conjugate a toxin, radioisotope, as I mentioned earlier, or even a failed drug, and use that as a carrier. An aptabody is a functionalized aptamer. So the aptamer itself it holds into position functional groups in 3-dimensional space. So it's a scaffold that holds functional groups in 3-dimensional space. So it's a supramolecular structure. Okay, so this is the next generation of aptamers. These aptabodies, as we coin the term. So with that, you can find very interesting properties. On this slide, I show, as we know, antibodies are protein-based. The chemical alphabet of antibodies are the 20 amino acids. Now, by definition, aptabodies can contain the 20 amino acids, but there's the possibility of using fatty acids, carbohydrates, organics, and metal ions. So it has a wide variety of applications, not only in diagnostics to improve affinities and specificities, but therapeutics in terms of improving pharmacokinetics or pharmacodynamics in terms of drug delivery and drug discovery in general, but also in industrial applications. So back to my model. If this is your scaffold, your naked aptamer, right? And let's say this represents like an amino acids, and an amino acid, a particular type of amino acid. And this represents let's say a small molecule, or maybe, small molecule or small organic, or maybe even a sugar. This represents maybe a fatty acid, okay? It's decorated with these functional groups, with the aptamer as a scaffold to hold these functional groups in 3-dimensional space. This is an aptabody, okay? This will have much more interesting properties than just the naked aptamer itself. Because of the chemical alphabet is much larger than that of aptamers and even antibodies, because we're incorporating amino acids, fatty acids, sugars. You can imagine a scenario where we're creating artificial enzymes that do not exist in nature. Okay, so we know that enzymes are protein-based, and most of the amino acids on these enzymes, these protein-based enzymes, is for architecture to hold into place a few amino acids that form a catalytic core or a pocket that converts a substrate into a product. Well, if you generated a large library of aptabodies, each one with a unique shape and decorated with different functional groups. And you interrogate that library, and you set up an assay to isolate enzymes that converts a particular substrate into a product, because you can imagine a molecular solution in that library that forms sort of like a catalytic core, right? That can possibly convert a substrate into a product. That's the possibility. That's where the field of aptamers are going, using these aptamers. We can convert possibly cellulose in as a substrate into some sort of biofuel, ethanol, or just let your imagination run wild in terms of using these antibodies in terms of industrial applications. But the key point here is that we're using, again, principles of Darwinian evolution on a molecular scale, and using chemical diversities to evolve these molecules to pass a certain fitness test, whether it's simple binding or as a simple nanobiotech application. So the strategy of separating winners from the losers or binders from the non-binders is also a key requirement for that. So this last slide is just a quick pitch that in terms of our company, we're a small biotech company in South Central Pennsylvania, as I mentioned earlier, and we do have employment and postdoc opportunities. And with that, thank you for your attention, and I can entertain any questions you may have. Thank you.
And thank you, Dr. Caltagirone, for that informative presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that this webcast will be available for on-demand viewing through May 2019. As a final reminder, Dr. Caltagirone will follow up with any questions you've submitted via email. That's all for now, and thank you for joining us. We'll see you again next time. Bye-bye.